So welcome back friends to another video in which we'll be talking about the structures of amino acids. So I'm Dipanshi Avasthi and let's get started. So friends, as in the previous video we have discussed about various amino acids and their types, their 3-letter code, their 1-letter code. So in this video I'll be talking about the structures. So I've already made the basic outline. So they all have this group in common, and the difference occurs only in the side chain which is attached to this alpha carbon. So in the glycine it's simply hydrogen H. As I've talked earlier, it is the simplest of all the amino acids because it only contains hydrogen, due to which its carbon is not the chiral carbon. You can say it contains two hydrogen, two same groups, so it can't be a chiral carbon, so it do not exist in the isomeric form. So we'll talk about it later. Then the second amino acid was alanine. So it contains simply a CH3 group in place of hydrogen. Then let's come to valine. It contains 3 additional methyl groups attached to the alpha carbon, CH3, CH3. Isoleucine and leucine, their structures are also somewhat similar. In leucine there are 4 carbons, 4 additional carbons. I am talking about additional only, so I am not counting this and this. So this is additional. There are 4 carbons, CH2, CH, CH3, CH3. This still isoleucine also contains the 4 carbons only, but there is only the difference in their arrangement. So they have same molecular structure, but these both are the kind of isomeric forms. So it contains CH which is attached to CH3, CH2, CH3. So they both have same number of carbons but difference only in their arrangement. Now let's talk about the methionine. As I've talked, it contains sulfur, so it contains 3 carbons. After 2 carbons there is presence of 1 sulfur atom, then it is further attached to 1 methyl group, CH3. Then the tryptophan. So tryptophan is a cyclic amino acid, you can say, because it contains the rings. It has 2 rings. The CH2 group is further attached to 1 pentose ring and 1 hexose ring. These are 4 double bonds. So this is 1 pentose ring and this is 1 hexose ring. So tryptophan, we can say in all of the structures, tryptophan is the largest amino acid. Its structure contains more number of carbon atoms. Then let's come to proline. It contains 1 CH2 group which is attached to another CH2 group, which is again bound to its amino group. So its structure is like this. Then comes phenylalanine. It is also a aromatic amino acid. These are aromatic amino acids because they contain at least one, you can say, 1 benzene ring or 1 phenyl ring. So phenylalanine. So to make it more simple, you can say it contains alanine. So see here the structure of alanine, 1 CH3 group. So here also it contains 1 CH3, but as phenyl ring is attached, so it will have CH2 and 1 phenyl ring. So remember the phenylalanine structure because this will be helpful when we will be discussing about the tyrosine structure, because it is similar to this phenylalanine, only the difference is at the para position there will be OH group present. All the rest structure will be similar in the case of tyrosine. So these are the structures of non-polar uncharged amino acids. So now let's talk about the polar uncharged amino acids. So serine. Serine is the simplest polar uncharged amino acid. It contains, as they are polar, so they will be containing the OH group at the terminal position. So it contains 1 CH2 with 1 OH. Then threonine. It contains 2 carbon additional, 1 CHOH and then 1 CH3. In cysteine there will be a single carbon only, but it will have one SH group, as I have told you that it contains the sulfur and it is involved in the disulfide linkage in the protein structure, so it contains terminal sulfur group, so that's why it is involved. One CH2 then SH. Now let's come to tyrosine. As I was talking about the phenylalanine, then I have told you to remember that structure. It is similar to phenylalanine, but in addition to this it contains one OH group. Rest the structure is similar. Then let's come to asparagine. Its structure is similar to aspartic acid, but the difference is one CH2 and then C double bond O NH2. Asparagine and glutamine, they are similar, but in glutamine there will be one additional methyl group present. It will contain one CH2, then another CH2, then C double bond O NH2. These both structures are somewhat similar, only one additional methyl group or CH2 group is present. Now let's move to our negatively charged amino acid, which is aspartic acid. So the aspartic acid's structure is similar to this asparagine. It contains one CH2 and then in the place of C double bond O NH2 it contains C double bond O OH. So it is negatively charged. So then when it is negatively charged, then it do not contains H. In place of it, remove this H and takes its electron, H become H positive, and it develops the negative charge. Similarly in glutamic acid, it contains CH2 CH2 C double bond O negative, because H is removed. So now let's talk about the lysine. So it contains 4 carbons. In addition, these are positively charged, so they will be containing the amino group at their end. Then it contains 4 carbons, CH2 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 CH2, and at the 5th position it will be containing NH3 positive group. That's why it is positively charged. Now let's talk about the arginine. So it contains the 3 CH2 groups. Its structure is a bit complex. 3 CH2 group. At 4th position there is one nitrogen, and then again means NH, then again there is carbon which is further attached to NH2 group, and with double bond it is attached to NH2, which is positively charged. So the structure is complex. There are here also there are 4 carbons, but after 3 carbons the NH group is present. Then with another carbon there is again another NH2 group, and with this carbon another NH2 group which is positively charged. So there are 3 amino groups, and here there is only one amino group after 4 carbons, and there are 4 carbons along with 3 amino groups. And the histidine, its structure is, it contains one CH2 which is further attached to it forms a pentose ring with the amino groups, carbon which is attached to NH. This carbon is double bondedly bound to one CH which is further attached to NH with double bond CH, and this NH group is positively charged. So this is the structure of histidine. It forms a kind of a ring, but it can't be aromatic because it do not contain the phenyl ring. So this is also a ring like structure, and this is all about these structures. So this is it for the structures. So there's another concept which I should discuss in this video only. So that was, I was talking earlier, that was about the isomers. So friends, to find the number of isomers possible for a particular structure, there is a formula which is 2 raised to power n, where n is the number of chiral carbons. And I hope you all know what is the chiral carbon. Chiral carbon is a carbon to which 4 different groups are attached. For example, if at one side there is a amino group and at other side there is a carboxylic terminal end, at one there is H, then R should be different. If it is, I think, let me take example of alanine, CH3, so that means here there is single chiral carbon, so the number of isomers will be 2 to the power 1, that will be 2. So there are 2 isomers possible for this. So let's get started from glycine. So it will have no isomers possible because there are no chiral carbons, because 2 similar groups are present to this carbon. Now let's move to alanine. Alanine contains 4 different groups, but this carbon has 3 similar hydrogen groups, so it can't be chiral carbon. So there is 1 chiral carbon. So 2 to the power 1 is 2. So 2 isomers are possible for the alanine. Then let's talk about valine. It contains, this carbon is a chiral carbon, so we'll put tick in this. Then we'll talk about this. It contains at one side hydrogen, but 2 similar methyl groups are present, so it can't be a chiral carbon. So it will also will have 2 isomers only. Then let's talk about the leucine. It contains this carbon, which is a chiral carbon. 4 different groups are attached, but when we'll talk about this carbon, it contains 2 similar hydrogen groups. Now let's talk about this carbon. It contains 1 hydrogen group but 2 similar methyl groups, so it cannot be chiral carbon. Then these two can also not be the chiral carbon, so there is only a single chiral carbon, so it will also form only 2 isomeric forms, so 2 isomers possible. So then let's talk about the isoleucine. So this carbon is a chiral carbon because 4 different groups are attached to it. Then we will talk about this. So this carbon contains 1 hydrogen group, 1 this group, and 1 CH3 and 1 CH2CH3. So this carbon is also a chiral carbon. This is not, and this is not. So it contains 2 chiral carbons. So according to formula, 2 to power 2 is 4. So 4 isomers are possible for the isoleucine. Now let's talk about the methionine. It contains 1 chiral carbon here. It can't be chiral carbon, it can't be, and after this the chain is broken, so it also can't be a chiral carbon. So it also will have 2 isomers possible. Then let's talk about the tryptophan. So this carbon is a chiral carbon. This can't be a chiral carbon as double bond is present. That means it can also not be, it can't be, and here also they also can't be. So it will also have 2 isomeric forms only. And similarly in the case of proline, it is a chiral carbon. It is also not, it is also not, and it is also not. So only 2 isomers possible here. Then in case of phenylalanine also, there is a single carbon. This is the chiral carbon. This can't be chiral carbon, and this benzene can also not be a chiral carbon. So it will also have 2 isomers. So in this you have seen that all have 2 isomers except for the glycine, because it do not have any chiral carbon, so it have no isomers possible, and the isoleucine, which contains the 4 isomers because 2 chiral carbons are present. Now let's move to the polar uncharged. So in the case of serine, we have 1 alpha carbon. This can't be alpha carbon, so here also 2 isomers are possible. And threonine, this is a chiral carbon. This contains 1H, 1OH, 1CH3. This is also a chiral carbon, so it will have 2 raised to power 2, that will be 4 isomers possible. Then let's come to cysteine. This is a chiral carbon. It can't be because 2 H2 are present, so that means 2 hydrogen groups are present. That means it can't be a chiral carbon. So in this also there are only 2 isomers possible. Now let's move to a tyrosine. It contains 1 chiral carbon. This can't be, so it will also have 2 isomers possible. Same in this case. This carbon is a chiral carbon. This can't be, this can't be. So only 2 isomers possible. Similarly in this case also, this one is only a chiral carbon, so 2 isomers are possible here. Now let's talk about the aspartic acid. It will also have 1 chiral carbon, so only 2 isomers. Then and here also this is only the chiral carbon present. It can't be, it can't be, it can't be, so only 2 isomers possible. Similarly in the case of lysine, whenever you see any CH2, then it's a direct, it's surely showing that it is not a chiral carbon. So here you can see there are only 2 isomers possible because this carbon is the alpha carbon. Similarly here also only the 2 isomers can be possible, and here also only 2 isomers can be possible due to this 1 chiral carbon. So in this whole structures, in the previous structures, you've seen that there are mostly two isomers only possible, because there is only a single chiral carbon in all, except for the glycine, which is not having any isomeric form because it do not have any chiral carbon, and the isoleucine and threonine. They both have two chiral carbons and they shows four isomers, and rest all shows two isomers. So in this way you can calculate the number of isomers possible for the other structures as well as carbohydrates and other biomolecules. So this is it for this video. I hope you have enjoyed it and you have learned a lot. So thank you friends, and have a nice day.